শুরু করে দাও।
ওকে, গুড ইভিনিং।
শুরু করব স্যার? Good evening, sir. So YouTube, everything is okay, perfect. Should I start?
Yes.
Okay. Good evening, everyone. The Department of Physiology and the Department of Sports Sciences, University of Calcutta, warmly welcomes all of you to the first episode of our webinar series. Our digital initiative waves the flag today on this very auspicious day as we introduce Physiosphere, the brainchild of our own department. The motive of launching such a digital space is to create a platform for the physiologists and by the physiologists. It is a stage for the proliferation of the subject physiology, the science of life. We want to join and hold all our hands together to make physiology a flourished subject. This is a forum for any and every individual across the globe who is associated with or wants to be related to the subject of physiology. In this era of digitization, the whole world is getting closely knitted, and so must the physiologists across the globe. This is our miniature initiative towards the attainment of this bigger picture. Physiosphere comprises of students, research scholars, and faculty members, also the staff of the Department of Physiology, and we shall be very delighted to help anyone who seeks information about this discipline. Physiosphere aims to create an association between the ex-teachers of the department, alumni of the Department of Physiology, and the existing students through such scientific webinars, exchange of ideas, and sharing of past experiences, information regarding the career opportunities, and certainly academic contents. We also want to help students explore various career options apart from the conventional professions and widen the subject's base and reach among high school students, the future, to attract them towards the subject. Physiosphere also targets to improvise the outlook of the existing students and scholars of the Department of Physiology in the context of the shifting paradigms and dynamic trends about the global scenario and status of the subject. The Department of Physiology and Sports Sciences of University of Calcutta requests all those members who have wandered through this sphere to share the glimpses of their life with those who are novices, the amateurs in this field. Now, the Department of Physiology head, Dr. Neelkanta Chakraborty, will speak about the department's legacy and the golden heritage.
Thank you very much. Am I audible? Am I audible?
Yes, please go
Yes,
ahead.
sir, you are.
Thank you very much. Most welcome, everybody. Good evening, Shubha. Hope all of you are safe and healthy. In this pandemic situation, COVID-19 pandemic situation, we have learned a lot. And arrangement of this program, this webinar lecture series, is our simple attempt to our academic approaches. The Department of Physiology that actually established in long before in 1902 in Presidency College, and the PG section opened in 1911, and that PG section was shifted to the Rajabazar Science College, where we are in Rajabazar Science College. The department is situated there on 1938. Lots of eminent teachers served this department, and at present we have 9 faculty members, 11 staff, and 40 more than 50 students in each semester. Uh, Most welcome to our inaugural speaker, Professor Dipak Sarkar from USA, one of our alumni of the department. In this regard, I must appreciate to to Professor Sanjeev Dey, particularly for his initiative to start this lecture series, and also our students who are actively participating here to organize the program. With these few words, I would like to start the program, and we should enjoy the talk of Professor Dipak Sarkar. Stay well, stay safe, and stay healthy. Thank you very much.
Okay.
Now, Professor Shomnath Gangupatay, the coordinator of the Department of Sports, will introduce our guest speaker for today, Dr. Dipak Sarkar, who's there in the Rutgers University presently. He's a prestigious alumnus of our department.
Good evening, but it is good morning in USA.
Yes.
So it will be my honor. It is my honor actually to introduce Professor Dipak Sarkar. And this is a brief biodata I have with me, but he has some uh, 39 pages biodata he has. So it is a brief of his whole career description. Dipak Kumar Sarkar received his D.Phil degree in neuroendocrinology from Oxford University in UK and Ph.D. degree in reproductive endocrinology from Calcutta University of course, in India. He is currently Director of Endocrine Programs and Board of Governors, Distinguished Professor in Department of Animal Sciences at Rutgers University in USA. He is studying how body stress and alcohol abuse program the neuroendocrine immune function and induces immune abnormalities, diabetes, and cancers. He is also working to develop therapeutic approaches for cancer and diabetes prevention. He has published over 200 research articles in high-ranked journals and reviews and co-authored two books. Dr. Sarkar has received numerous awards, including state scholarship from West Bengal government, S.K. Manchanda Memorial Oration from FIPS, Shubha Mukherjee Memorial Oration from our very own society, Physiological Society of India. He is the Fellow of Indian Academy of Neurosciences, Fellow of American Association for the Advancement of Science and National Institute of Health Ameliorates Award. Recently, he was awarded 2017 Distinguished Researcher Award from Research Society on Alcoholism and 2020 NJ Academy of Science Inaugural Advancement of Science Award. Today, his topic of lecture is neuronal activation of endorphin. Very interesting. Neuronal activation of endorphin ameliorates aging-induced hyperglycemia and insulin resistance. So now Professor Sarkar will deliver his talk.
Okay, I'll share my screen first. Right. Okay, can you see my screen?
Visible?
Yes.
It was visible.
Uh, now it is black. Maybe
Yeah.
you just show any slide or start your presentation, then it will be shown. Now the screen is black.
But I'm moving, it's not.
Uh, yes, now it is. Now it is on the screen. Yeah.
Oh, it's showing. So when I get a full screen, it doesn't go. Let me see. Is the screen moving?
Yes, of course, it is moving.
Okay.
Of course,
So
it is moving.
what
Yes,
about this way? Full screen.
you just full screen. Make it full screen
Yeah.
from your
Is it
in your
moving?
computer.
Okay.
No, it is not coming. It is again. It is black.
Okay. So full screen doesn't work. All right. I'll go with this. That's okay.
No. Can you please that in your laptop? Can you make it the full screen or presentation, full screen presentation from the laptop? It
When
will
I
work.
did that, it's coming out black over in there.
Okay.
So
Okay.
this is the only way you can see. I could.
Okay, no problem. No problem. Then I may request the Shoti to enlarge the screen so that the whole screen will be covered by the yes. Now it is okay.
Now it
Now
is okay.
please
Absolutely.
continue. Yes.
Okay. All right.
Yes.
So thank you very much, Sandeep, and thank you, Professor, all the faculty from the Calcutta University Department of Physiology, and all the students for inviting me. I think it's my very good pleasure. I was at APC Road, Calcutta University, from '68 until 75. And Dr. Moito was the chair at that time, and physiology was in a very small group of colleges and university, and many of us under Dr. Moito, we went to Diga, walking, spreading the importance of physiology, went all over the state, and was able to then open Vidyasagar College as honors, and then several other colleges now is spread out, and it's very good to see that physiology has become a very important subject area in Calcutta University. So it is when I heard that I was invited to give this inaugural talk, I was absolutely pleased to give this. So it's my great pleasure. And I also selected some good topics, which is most of the things which we haven't published. So a very cutting edge research presentation, because I want to give that to my own department. So that's what I selected. I also selected an area which is very relevant to India as well as to many other part of the world, which is diabetes area, and also wanted to present something very novel, very exciting, which may identify that there may be a novel therapeutic approach to prevent this disease. So let me come to showing the diabetes. What is diabetes? When you say diabetes, we really mean diabetes mellitus. There are two kinds of diabetes. One is diabetes insipidus, another is diabetes mellitus. What is diabetes insipidus? Diabetes insipidus is the one when the hypothalamic vasopressin neuron doesn't work, so body cannot absorb the water, so we have continuous urination, heavy drinking, heavy urination, and that is the major problem with the diabetes insipidus. Diabetes mellitus, on the other hand, is a problem with the glucose. As you know, when you eat rice and other carbohydrate molecules, it breaking down, forms glucose, and the glucose. So that's good thing. So why is it a problem? because it is a problem, it cannot get utilized very well. Glucose gets stored in the muscles and the liver, and it gets stored as a glycogen. And when you have a diabetes, it cannot get stored, it cannot break down easily, so the blood level of glucose goes up. And what is the big deal with that? Big deal with that is when blood get heavy in large, there is a small vessel in the eye, and that breaks down, and that's called retinopathy. Then because of the glucose it makes this blood concentrated, you get a neuropathy, you get a pain in your back and your feet. And also because of the thickness, you also have a kidney problem, because kidney cannot clear the agent very clearly, so you get a diabetic neuropathy as well, nephropathy as well. And also at the same token, there is a risk for heart disease, and stroke also arises. So it's not a really simple thing. It can be controlled very well, but if it's not under control, it could cause a major problem. And in general, it cause a lot of death in India and throughout the world. So what are the type of diabetes? There are three types of diabetes. Diabetes type 1 diabetes, when it really happened, is called another called insulin dependent diabetes, is called, and also happens in the juvenile onset, and that's why it's also called juvenile onset diabetes. And it begins in childhood, particularly when you have a subject or patient with an autoimmune condition, and that autoimmune condition destroy the pancreas, prevent the insulin secretion, and that leads to the diabetes. And maintenance of the insulin in this person is very critical. Without insulin giving to them, it could bring a lot of pathology and death. Type 2 diabetes, on the other hand, is really, it happened varieties of reasons, but it's primarily insulin is maybe there, but insulin doesn't work. Why it doesn't work? Because the system, various tissues and all those cannot respond to insulin. So you get insulin resistance. Also, sometimes one of the cases I'll show you here is aging, when the pancreas doesn't produce very much insulin, and therefore you get also low amount of insulin, a large increase of blood glucose, and the resultant of diabetes. And finally is the gestational diabetes. It happens, some patient, not always, during pregnancy. So pregnancy induces this, the insulin resistance, and that could lead to some form of diabetes. Again, it's required control, and because it's happened during pregnancy, it's called gestational diabetes. So these are the basic three diabetes, but the major diabetes are type 1 and type 2, that's affecting throughout the world. And that's what's shown in these slides. Here you can see the red area is an area where there's lots and lots of diabetes. And within the red area, India falls in it. And there are about 382 million people with diabetes in 2013. Now there's additional 20 or 30 million increase. So it's over 400 million diabetes patients throughout the world. So it's a major, major problem of India. And believe it or not, 50% of the people with diabetes lives in Asia. Almost one up in China and India combined. So India and China is a major problem. And there's currently about 62 million Indians, which is more than 7.1% of the adult population, has diabetes. Average age of the onset of diabetes is about 42 years, and nearly or more than 1 million Indians die due to diabetes every year. So it's not only in India, it's also a problem in US. 29.1 million people in the United States have diabetes. 8.1 million maybe, diabetes is not easily recognizable because there is a pre-diabetic condition. Many people do not care they have this pre-diabetic condition, because that condition leads to the diabetes. And there are many people even in India and US, have this pre-diabetic condition. And more than 1 in every 10 adults who are 20 years or older has diabetes. For seniors 65 years and older, that figure rises to more than 1 in 4. So aging is a major problem. So adult age 40 to 49 comprise the world age group with the highest diabetes rate. So when you look at the aging population, particularly type 2, is really large and is a very high risk for older age is a very high risk for causing diabetes because of the age which alter metabolic function, which alter many hormonal control, and that is a major age for diabetes, initiation of diabetes. So aging is the one of the normal cause for diabetes. So we thought we use the aging model to see whether or not we can understand the process of diabetes, or we can develop some treatment which can prevent diabetes. So let me, as you can tell from my title, that I'm going to deal with the brain. I'm a neuroscientist. I'm an endocrinologist. Neuroendocrinology my major focus, but I also work cancers and many other diseases. So let's see what is the brain. How does the brain participate in controlling glucose metabolism, and how this function can lead to the initiation of diabetes. So this is a sagittal section of the brain. As you can see, you see the olfactory bulb is a rat brain, and this is the area hypothalamus. Here you get the medulla, pons, and midbrain, and so the nerve, sympathetic, parasympathetic nerve, goes from the hypothalamus, enter into the peripheral nervous system, and within the hypothalamus, this area, there are certain area of the nucleus of the area. There are lots of neurons which are very sensitive to glucose, and they also participate in regulation of glucose metabolism. Particularly important ones are the area of BMH and DMH and arcuate area. Within the arcuate area, there are neurons called AGRP, NPY, proopiomelanocortin, POMC, and SF1. SF1 is steroid factor binding 1. These are all glucose sensitive, and they also regulate glucose metabolism. So how do they do that? So let me show you one of the example of POMC neuron, proopiomelanocortin neurons. And these proopiomelanocortin neurons are very glucose sensing, and they are very much involved in preventing food intake behavior. How does it do so? It's when it's get activated, it produces alpha MSH. Alpha MSH then goes and act on the melanocortin neurons, and then that prevents the food intake behavior. And is also get regulated by, is also prevent NPY neuron, which cause increase of food intake behavior. And POMC neurons are sensitive to glucose. So when glucose comes, glucose enter into the blood through the median eminence or through circumventricular organ into the brain, passes the blood-brain barrier into the brain, and once it enter into the brain, is get taken up by the glucose transporter, and glucose transporter then is entered into the neurons, and there you have a glucose kinase. Glucose kinase then metabolize to produce this glucose to ATP. Once the ATP is formed, it activate AMPK. AMPK is the one which activate the neuronal potential and neuronal activity. And also, glucose can also enter through the astrocytes. Astrocytes then metabolize it to a lactate, and lactate then enter into the neurons and then also produce ATP. So ATP, once the ATP is activated, it activates the potassium ATP channel. Potassium ATP channels then depolarize the neurons and activate the POMC neurons. So the POMC neuron activation goes down when AMPA-K reduces, and it reduces the glucose, the POMC neuronal activation. So POMC neurons is quite important for regulation of glucose. When glucose goes up, POMC neuron goes up, and then it prevents the NPY neurons say don't eat anymore, and it stops, and glucose level get balanced. So POMC neuron is very critical. So again, putting that in cartoon, showing how POMC neurons control the glucose and feeding. So here's the POMC neurons is producing alpha MSH, is activate the MCR receptors on the neurons, and then it prevents the food intake behavior. And POMC also act on the NPY neuron, prevent the food intake behavior, because NPY causes the food intake. And NPY get activated by ghrelin. Ghrelin comes from the stomach. When you are hungry, the stomach contracts and produces ghrelin out. Ghrelin goes to the brain, activate the NPY. NPY then immediately say, oh, you're hungry, go and eat. So NPY then initiate the cause of eating. And when the ghrelin goes down, insulin and leptin at CCK. CCK is the one of the hormone which gives you satiety when you have enough feeding, and insulin also rises because the glucose is activating the insulin production from pancreas, and that turns this thing off. Activate the POMC, turns this thing off. That's the way food intake, blood glucose, insulin get all maintained. So one of the things happened. Before I go there, one of the things happened is that this POMC neuronal number is shown by us, by various others, that goes down during aging. So we hypothesized that if POMC neuron goes down during the aging, and that maybe if they are involved in glucose control mechanism, can we activate POMC neurons by putting some POMC neurons or bringing some novel mechanism to activate them? Could he prevent that aging-induced hyperglycemia or diabetes? So that's the hypothesis we are testing here. So how do you do that? Well, before this is a very interesting novel technique. We started working on. We have a recent paper got accepted in Endocrinology. Will show some of those data, and also we have established this novel model I'm going to describe. So let me introduce with the stem cells within the brain. So this is a picture taken from the work of Kai et al. What he did, he made these transgenic mice, put the GFP into the nestin. Nestin is a transcription factor which identifies stem cells. He put the nestin, the GFP on nestin, and make a transgenic mice. What he found, there's a large number of these nestin positive cells in the lateral ventricle or subventricular zone of the brain. And he also found there's a lot of nestin positive cells in the third ventricular area and also in the pituitary. So suggesting there's a lots of stem cells, neuronal stem cells or stem cells within the hypothalamus, even the third ventricle. So we thought, oh wow, okay, so there are there are stem cells within the hypothalamus. Can we somehow modify these stem cells and make them beta endorphin neuron? So we tested the hypothesis. So Natka Vajiva is senior research fellows in my lab, and I asked her to test this out. So what she did, she made the stem cells from the fetal brain and stem cell, then put these molecule like cyclic MP and PCAP. PCAP is a peptide in the pituitary which produces cyclic MP activity, cyclic MP-like substances. So with this activation of neuronal stem cell, it shows there's a beautiful lots of production of beta endorphin neuron. This paper has been published in Proceeding of National Academy of Science, and then she saw that the cyclic MP cap produce these stem cell into beta endorphin cells at various stages and makes 100% beta endorphin neuron. So then we put this beta endorphin neuron in the hypothalamus within the ventricular, within the PBN, where the beta endorphin neuron terminates as well as make connection with the autonomic nervous systems. And we find this when you put them in the, transplant them in the PBN area, they remains there, remains for months and months. And beautifully, they make all this neuronal connection. And more excitingly, they act like normal beta endorphin neuron. How do we know that? We challenge them with the stress. We find these more beta endorphin neuron we put in, it suppresses the stress response more and more. So it is a beta endorphin neuron suppresses a stress function. It also activates immune function. So this is the paper published in PNS in 2008. So we discovered this many years ago. And then, but we have published a lot of things. I wanted to patent this, but the pharmaceutical company said, how are you going to put these endorphin neurons within the human? Where are you going to get this fetal brain? And how are you going to make these endorphin neurons and put in the human? It's going to be very difficult. So we didn't get depressed. So we came another method. How we did this? We made this nanosphere molecule. Nanosphere molecule, you can put them, inject them in, and they can go all different places. Even they can cross the blood-brain barrier. And we attach this nanosphere molecule with cyclic MP and PCAP and looked in the first in cultures and found that they releases the beta endorphin neuron within the culture. Then we injected into the third ventricle, and we find is more cyclic MP they are, more beta endorphin get produced. This is a beta endorphin and BRD staining. This is a control animal showing the BRD are not attached with beta endorphin. And here, look at this. More cyclic can be put. This beta BRD or novel neural new neuron making beta endorphin, showing again here new neuron making beta endorphin. More you put in, more beta endorphin is produced. So this technique has also got published 6 years ago. And then what we using those two techniques, we found a very exciting finding, which is we found that activation of beta endorphin prevent prostate cancer, breast cancer, pituitary cancer, lung cancer, liver cancer, colon cancer, all in animals, but all these cancer got prevented. And they're published in all those top-notch journal, Cancer Research, PNS, all those journals. There are about 9-10 papers on this. So, and we are still working on trying to see whether we can take this to human this activation process and some of the signaling cascade and prevent some of the cancer in humans. So now we have a grant working in using human patients for cancer prevention by the pharmaceutical compound which produce the beta endorphin activation process into and activate immune system function. So the question, will come into your mind and others that how does beta endorphin in the brain preventing cancer, preventing all the other stuff? Well, interestingly, what does beta endorphin do? When you put the beta endorphin there, it altered autonomic nervous system function. It activates parasympathetic neuron and suppress sympathetic neuron. And how do you know that? And this is the cover page of the cancer research. We showed that by beta endorphin, we can prevent them by naltrexone, which is an opiate blocker, prevent this effect. And we prevent this effect by preventing the parasympathetic neuron with another blocker, and sympathetic neuron and activate sympathetic neuron by metprotinols and others, showing that this is caused because of the activation of parasympathetic neuron and sympathetic neuron. So we have a model. Let's see whether it works in the aging and whether or not it works in diabetes. All right. So why did it come to this concept? What it did, the question arises: if we activate beta endorphin and leave the animal or the patients for a long time, what happened with this excess endorphin? Okay, does it make any problem for the body function? So we did some study to show that this beta endorphin neuron activation really doesn't cause any alteration of body function. How do you know that? Well, we did all different study. One is we looked at the aging animal, their circadian rhythm, their biology, their running wheel activity. What do you find? That this aging reduces this running wheel activity, and when you put these beta endorphin neurons and leave them for a long time, their activity goes up, so they become more younger. Another interesting thing, this is the rhythm gets better, and another interesting thing, their activity goes up, so they feel much younger. And look at this: the body weight slightly reduced, not very much, but it's reduced. The glucose goes down. Glucose, there's a aging-induced normal glucose. It goes down. So we thought, oh, this is very exciting. So this could be something good for diabetes. So also we looked at the heart rate, blood pressures, and liver weight, spleen weight. Nothing we found very significant. We found pituitary weight in animals. Pituitary weight grows and it develops tumors, but beta endorphin neurons suppresses this. We have a grant working on this aspect too. So this will be the aspect of my talk today, and I'm going to show you that the beta endorphin treatment, neuronal activation, also can be a very good way to prevent aging-induced diabetes. So let's do some experiment and see where we go. So Ali al-Yassari, he was from Iraq. I got an Iraqian fellowship and came and worked with my lab. And what he did here, he used to check the concept that whether or not this beta endorphin neuronal transplant have any potential preventive effect in diabetes. So in animals, diabetes can be used by a chemical called STG. STG, what it does, it really destroys the pancreas. It causes like type 1 type of diabetes. Destroy the pancreas, destroy the insulin producing cells, and elevate the blood glucose tremendously. Here, showing this, when you treat the animal with STG, your blood glucose level achieved is 450, nearly 4 to 450 milligram per deciliter. It's very, very very high level of glucose in the blood. And POMC neuron, we did for a short time, and it significantly reduced this blood glucose. Not completely, because STG in this case is really made this diabetes absolutely going to the roof. So it significantly reduced. Another interesting thing is that blood insulin level in aging STG animal goes down because is destroying all the pancreatic islet cells which produce insulin, and it's reducing insulin. And POMC is appearing to be increasing the insulin level, and also the insulin receptors level. It gets elevated during the STG because it's really trying very hard to make the glucose normal, and POMC transplant suppresses it. So let's see what happened with the structure of the pancreas. Within the pancreas, you see the islet cells, you see the control animal, islet cell producing lots of insulin. With the control STG, look at this, islet cell got basically depleted with the very little amount of insulin left. POMC treatment, getting those islet back to normal. Not completely normal, but back to produce more insulin producing cells. Glucagon level also reduces STG. It does not so much like insulin is really get affected by STG, and glucagon also does, but the POMC cells also has a beneficial effect, but not very significant. So POMC neuron also this is also showing the program death. The pumps, the islet cells, they have lots of cells are dying, and POMC is preventing this program death. So basically, POMC neuron activation is preventing the death of the islet cells which produce insulin. The caspase-3 activity, which indicates the death of the cells, and STG increases this. POMC transplant suppresses this. ROS is a oxidative stress marker which get activated. You get a more cell death, and this ROS production goes up in the STG, and it prevents by POMC. So basically, showing POMC activation has a beneficial effect on the pancreatic islet of Langerhans cells, which produce insulin by preventing the reducing oxidative stress and preventing program cell death. Also, it changes cell proliferation. In STG cells, there are slightly increase of cell proliferation because cells are all dead. So body has a mechanism to try to increase the cell growth to prevent this STG-induced death. And but when you have a POMC, there's a marked increase. So, and that's really bringing back some of the islet cells to produce insulin. So this Ki67, one is the cell growth marker, and CDK4, another growth marker, both shows that the POMC neuronal activation really cause increase of the cell growth within the pancreas producing insulin. And how does it do that? So one of the hypotheses we have that it maybe do so because it reduces the inflammatory environment. Because, the diabetes condition, you have a lot of inflammation, body inflammation, not only diabetic condition, even in COVID, inflammation is a major problem. Inflammation is the one which, causing all those problems. And interestingly, the beta endorphin activation is really reducing. I will show you in this model as well as the aging model is really reducing inflammation. Here, showing IL-1 beta is a inflammatory cytokines going up with the STG, POMC suppressing it, TNF alpha, another inflammatory cytokine going up with the STG, POMC suppressing it, IL-6, another inflammatory marker, not going up so much, but POMC has a slightly beneficial effect. IL-17 not going up so much. IL-10 is an anti-inflammatory marker, which goes down with the STG, and POMC is increasing it. So POMC activation causes the alteration of this ratio, inflammatory and anti-inflammatory. So it's increasing anti-inflammatory cytokine, decreasing inflammatory cytokine, making it is a more immune responsive subject. Also, POMC is shown with the other inflammatory cytokine, PRF1, granzyme B, interferon gamma. These are all in spleen. The spleen is another area where the most of the immune system works. Within the spleen, granzyme B, perforin are the two cytotoxic agents. They go and kill all the bacteria, virus. They get activated by POMC. Interferon gamma, also produced from the NK cell in the spleen. This get activated to go and kill the bacteria, viruses. IL-2 doesn't change. IL-6 slightly suppresses because it's the inflammatory cytokines. IL-17 doesn't change. IL-4 is a anti-inflammatory substance, goes up by POMC, and IL-10 also anti-inflammatory substance goes up. So basically, as I said, this POMC activation really preventing the inflammatory environment in the targeted tissue like pancreas and immune reservoir like spleen, where the ratio of anti-inflammatory inflammatory is changing and making them less inflammatory environment in the body. All right, so our system, the beta endorphin is working and is working with the STG model. Let's go into the aging model. And this work is primarily done by Dr. Shani Mukherjee. Shani is also from Calcutta University. She worked in Science College as well as the Colorado Institute, and she came and joined after she finished her PhD. And what she did now, she used the aging models. These aging models are at rats about 12 months old. And what she did, she has this control, which is just the control aging, control young, beta endorphin young, control old animal with the control transplant, old animal with the beta endorphin transplant. What you see here, that plasma glucose goes up when you get age, and then beta endorphin neuron suppresses this, bring it normal. Insulin concentration also slightly goes up in the aging, slightly goes down in aging, and beta endorphin transplant reverses this. Insulin receptors, also beta endorphin, the insulin receptor aging goes down, and beta endorphin transplants bring it back. So just like the STG, this STG had less effect with the STG level. Glucose was humongous, but aging animal, look at this. Each time glucose, insulin receptors, they are coming back close to the normal range. So aging-induced this beta endorphin transplant is really making it back to young life, normal young level. Also, this is the beta endorphin level. This is the normal control young pancreatic beta endorphin. This is the aging pancreatic endorphin. This is the aging beta endorphin transplanted pancreatic beta endorphin, basically to very much normal, shown here. So in the aging system is working really beautifully and bringing back to the youth kind of environment, as we have shown before in respect of the activity rhythm, diurnal rhythm, all those other things is really turning very youth. So continuing that about this neuronal activation in aging rats, here she studies the inflammatory cytokine. TNF alpha is one of the inflammatory cytokines. Aging animals in the pancreas, the inflammatory cytokine is going up. The beta endorphin transplant bring back to the normal range. IL-6, another inflammatory cytokine going up in aging, beta endorphin is bringing back to normal range. It is IL-1 beta, another inflammatory cytokine, interferon gamma, they are all going up, inflammatory cytokines, and beta endorphin transplant bringing it back to normal. Caspase-3 in the pancreas, caspase-3 is a marker for the cell death. Cell death, as I showed before, the pancreatic islet of Langerhans cells were low, small, because the cells were dying, and the caspase-3 shows that cells were really dying, and beta endorphin transplant bring back to normal, prevent this cell death. CDK4 is another cell growth marker. Cells were dying, and then the beta endorphin transplant activate this cell growth and bring back to normal. IL-10 is the inflammatory anti-inflammatory cytokines, goes down in the aging animals. Beta endorphin transplant bring back to normal. So basically, just like the way we saw in the STG animal, is doing the same thing, but is doing better with STG, like STG, which really destroy the whole pancreas. Here is not completely destroyed. They're still remaining, and beta endorphin neuronal activation now making that back to the young life and bringing back to the normal. Is also anti-inflammatory cytokine gene in spleen. As I showed you in the STG case, spleen is a really immune reservoir. Here also, IL-6 is an anti-inflammatory, with aging going down, with the beta endorphin bringing back up. 17 is the reverse, is anti-inflammatory, going up, reversing. IL-2 is not going up very much, but beta endorphin preventing it, making them most anti-inflammatory. IL-1 beta going up, and beta endorphin suppressing it. IL-4 going down, which is a anti-inflammatory. This is inflammatory, is reversing it. So just like very much like the STG, even in aging model, is really making the body immune system more beneficial, more functional, because the inflammatory cytokines levels are down, anti-inflammatory cytokines, good cytokines levels are up. NK cells, which is a cytokine, which are innate immune cells, they are the one which first attack the virus and bacteria. They are the one getting activated by beta endorphin, really, making those the body immunity very strong. So immune system goes up, goes better, just like the way we saw with the cancer, and this system, here we are seeing the same thing. But does that, is it impacting the insulin effect on the tissue, just like in the liver and other place? We also tested that, and we looked at the insulin receptors in the liver, which goes down in the aging. The beta endorphin neuron activation brings back to normal or activate more in glucose phosphate, which are really get altered, because glucose phosphate is the one which is causing causing more activation of production of glucose, and that's getting activated by the aging, and beta endorphin is suppressing it. Pepsi K, another the glucose metabolism regulator, which is altered in aging, and beta endorphin trying to bring back to normal. FOXO1, glucokinase. The FOXO1 is another glucose metabolic activator, is increasing and is suppressing by beta endorphin. Glucokinase reverse action, which is suppressing in aging, reversing back with the beta endorphin transplant. GLUT2 receptors also is getting reversed by beta endorphin transplant. So if we can put them together, what we do see that within the liver, in the normal condition, when the glucose insulin receptor get activated, it activates the RS, P13K, AKT1, and that cause the glucose uptake, as well as formation of glycogen in the liver. When in the aging, this glucose system get altered because of the alteration of the P30K, AKT pathway, and this really mess up the insulin response, and the glycogen production and glucose uptake severely defective. With endorphin transplant, on the other hand, is really reverses it back, even within the liver. Perhaps this is caused by probably the inflammatory environment or many other mechanisms which we are not sure. But beautifully, beta endorphin neuron also act on the insulin receptor cascade within the liver, and brings them back to normal insulin receptor function. So again, the question arises, we are putting beta endorphin transplant within the brain. How is it so, making these back to normal? So we use these blocker of sympathetic neuronal activity, metprotenol, and when you do that, this is a normal young animal blood glucose level following glucose tolerance test, and then we're in the aging. This is the level quite elevated, and glucose is very high. When you do beta endorphin transplant, the aging animal goes back just like the young animals, but when you block them with the sympathetic activators, they go back, behave like the aging animal, showing the sympathetic nervous system is very critical for or prevention of the sympathetic activity is very critical for the beta endorphin neuron activation. So basically, beta endorphin neuron, when you put them in the brain or make the beta endorphin activation by the nanosphere or by beta endorphin neuronal transplant, it suppresses the sympathetic neuronal activity and activate the parasympathetic and cause the prevention of inflammation and cause the alteration of the normalization of the hepatic function and increase of the Langerhans islet of Langerhans production of insulin, and making them back to the normal like young phase. So in conclusion, then This data which I presented there, none of them are published. We are in communication. So I think this will be very exciting. Hopefully, we can take this to, in the future, some development of new drugs or something to prevent diabetes. But these data I showed you today indicate that the POMC neuronal transplant or activation reduces hyperglycemic condition and improve insulin function by decreasing pro-inflammatory environment and preventing pancreatic pathology by modification of autonomic nervous system function. And finally, even though I showed the people who were involved in this study, but there are many others there who are also in the lab, and they're participating, help some of the finding here, and also participating with other studies. We also have graduate students and undergraduate students also participate in this research, and our research has been funded since I have been very lucky. I have received funding since '83 and continuous funding, and from the National Institute of Alcoholism, as well as the Cancer Institute, and also the Child Health Institute. So with that, I want to complete my presentation, and thank you for listening, and I'm open for any questions. Appreciate your invitation. Hello?
Now we'll see if there are some questions from the audience that can be conveyed to Sir.
Okay. Okay.
Yes, sir. There are a question from Vishwadev Bishai.
Okay.
How inflammation or inflammatory cytokines activates POMC neurons as opposed to diabetes?
So I haven't presented any data showing the inflammatory cytokine activate POMC. My data shows that POMC neuron activation is really increasing the anti-inflammatory cytokine level and suppressing the inflammatory cytokine levels. And that is being done because it's a complex mechanism, but because POMC neurons reduces the stress then locally alter the sympathetic, like splenic function. Spleen is the reservoir of varieties of immune cells, NK cells, T cells, B cells, dendritic cells, and they're the one producing these cytokines, inflammatory or anti-inflammatory cytokines. So by altering the stress regulation, it also have a control over the immune system to modulate the inflammatory and anti-inflammatory cytokines. And when the stress level goes up, the stress hormone then goes into the brain, and then the beta endorphin level goes up, and then beta endorphin suppresses stress level to maintain the body immune function. Did it answer your questions?
Any other question from the audience?
Yes, there is another question. What is the suggested link between POMC neurons and autonomic higher center?
POMC neurons are connected with these autonomic nervous system. How is it connected? POMC neuron terminate in the area called paraventricular nucleus of the hypothalamus. Paraventricular nucleus then sends another branches of neuron to the lateral hypothalamus and NTS area, which then carries all the way to the spinal cord and distribute or connects with the autonomic nervous system to distribute all the peripheral organs. So, is interestingly, both POMC neuron as well as CRH neuron has a direct control over autonomic nervous system. What we have shown, this study and many other studies, that POMC neurons activate parasympathetic activity and suppress sympathetic neuronal activity through these neuronal cascades.
Any other questions?
No, there is no question.
Okay, so if there are no more questions from the audience, then I would like to thank Sir for conducting such a nice session. It was really very informative, and I hope the audience enjoyed it a lot. And now I'd like to introduce Professor Shonjit De from our Department of Physiology, who has actually conceptualized this whole thing of, initiating a sphere called Physiosphere and initiating an online platform that could actually bring together the physiologists. And it has been really wonderful. So I would just like to say that, sir, you may now start by saying whatever, the vote of thanks and all.
Okay, so good evening, everyone, and good morning to our the audience at the Western Globe and the other side of the globe. And my thanks and appreciation, my gratitude from the bottom of my heart to everyone for participating in this webinar and for today's talk and and participating in this Physiosphere. And okay, I'm coming to thank Professor Sarkar later, but before that, since it is my pleasant duty, the Department of Physiology and the Department of Sports Science, University of Calcutta, unfolded the globe or the sphere for the physiologists by the physiologists today in this holy day of the Navaratri, when Ma Durga has entered the globe to ward off the negative energies, bringing health and peace to the world in this pandemic situation. So it is, apart from any religion, it is, I think, she will bring peace and harmony and also the good health to everyone. It is my pleasant task to extending vote of thanks now. And so I must profusely thank our Honorable Vice Chancellor, Professor Sonali Chakraborty Banerjee, Pro Vice Chancellor, Academic Affairs, Professor Ashish Chattopadhyay, and Pro Vice Chancellor, Business Administration and Finance, our Registrar, Professor Devasis Das, and Dean of Sciences, Dr. Tanuka Chattopadhyay, Professor Tanuka Chattopadhyay of my university. And also I extend my thanks to Science Secretary for his all-time support. And my best appreciation for their motivation and support to my head of the department, Professor Nilkanta Chakraborty, who actually formally inaugurated this Physiosphere. And in spite of all his administrative business during this, daunting task of, arranging examinations and all, he gave me all support and also extended his, good wishes for all to extend this event all the way. And Professor Somnath Gangopadhyay, he is always a good source of support and encouragement to me, and in all time of need. And I cannot belittle his support as as a big brother. We, the community of the Department of Physiology, and with the bunch of motivated second semester students, and with our energetic mentor faculties and the staff, we started this journey of the Physiosphere. It is needless to mention that we work as a team, and we share the support and from our senior faculties like Professor Devashish Bandyopadhyay. He is my teacher also, and my friend and my colleague, Professor Bishwadeep Bishoy. However, I must appreciate the contributions and the positive organizational efforts, particularly from Rajan Haldar, who brought the reunion of our department into the front stage once more. And we hope that we will continue it again. Dr. Amit Bandyopadhyay, Dr. Roshnara Mishra, Dr. Shreya Chattopadhyay, they are always being so supportive, and they are in true words, they are instrumental in motivating students and uplifting the spirit of our department. So a big kudos to all of them. Well, and I should not belittle now belittle now the Professor Sarkar, for Professor Dipak Kumar Sarkar, for agreeing, for extending his lecture and warmly accepting our invitation. He has enormous contribution in his field and in all niche areas, in very critical areas of the physiology. And I think that is a wonderful, learning point for the, future physiologists. So that's why I invited him. Okay, just for the future physiologists. Okay, so you can do whatever you can. And he has enormous, contributions in the physiology and the animal sciences, making this our university, our department, on the physiology fraternity enormously proud. And on the words of appreciation on behalf of the department are too minuscule to thank Professor Sarkar. I'm really honored, sir, to host you. And I'm positive that he will keep us giving his valuable support to our department for all constructive causes and for the future footsteps. He always remains humble to his department. Perhaps he has a big heart always, and it beats with his alma mater. And okay, and I must say that, sir, huge thanks from the department. Though you are, our proud son of our department. Okay. Though we lost recently some 3 great luminaries from our galaxy of good souls from this department. We lost Professor Vishwanath Kole, Dr. Umar Ganguly, and Dr. Pritin Banerjee in this pandemic time. However, we do wish to carry out their dreams with our positive work for the society. And essentially, we hope that this digital initiative waves to last long, and with creative ideas which will adorn the physiosphere, making it a colorful flower, it will blossom. And the major purpose of this physiosphere is to tie up with the society, to reach for those who need actually, okay, and for their pressing demands. And essentially, well, some of my friends, they have asked, sir, whether we should register or whether we should get certificate. Okay, so don't make it a, those things, very formal. Actually, it will remain as a reunion of the physiologists. And in the name of the academics, though, we will not make any boundary in this teaching learning initiative. All right. And with this philosophy, we invite everyone to send opinions and the constructive criticism so the physiosphere can blossom in its own way. All right. Essentially, we will go ahead with our tributes to our teachers, with their interviews, with their contributions, with their sharing good thoughts in our department, okay, and for which we enjoy the subject today. Okay, my hats off to all these teachers. And well, we will now like to watch 2 short promos of our beloved teachers. So just please keep on watching these promos. Okay, after my vote of thanks. Thank you very much once again for participating. I hope that this will, give you solid support. Your participation will give us a solid support. Okay, so just watch this promos. Thank you very much once again. Is audio coming? Okay, Physiosphere team, audio is not coming. Okay, should I run from here?
Audio coming? Okay, sir, you can run from there. There is maybe some technical glitches.
Yes, sir, you can run from here. Like from your system, because I think there are some technical glitches.
তো আমি এই উদ্যোগটা নিয়েছি এই কারণে যে আপনার এই আমাদের শিক্ষক জীবন সম্বন্ধে বা শিক্ষকতার আপনার অভিজ্ঞতা এটা যদি আপনি কিছু শেয়ার করেন, তো হ্যাঁ, তো আমি আপনাকে জিজ্ঞেস করছি যে আপনি যে শিক্ষকতা এলেন তার মোটিভেশনটা কি ছিল?
আচ্ছা, আমার এই টিচার হওয়ার মোটিভেশন সম্বন্ধে যদি কিছু বলতে হয় তাহলে স্কুল জীবন থেকে কলেজ জীবনে ঢোকার পর কিছু কিছু অভিজ্ঞতার কথা বলতে হয়। আমি চার্চ কলেজের স্কুল থেকে স্কুল ফাইনাল পাস করি, বুঝেছো? এবং আমাদের সময় তো তখন স্কুল ফাইনাল এক্সামিনেশন ছিল, হ্যাঁ, 10 ক্লাসের পরে আর কি। সেই সময় স্কুলের আমাদের হেডমাস্টার ছিলেন একজন ব্রিটিশ, জেসি হেন্সম্যান। উনি খুব ডিসিপ্লিন ছিলেন এবং খুব পাঞ্চুয়াল ছিলেন, এবং ওনার একটা খুব সুন্দর ইমেজ, একটা সুস্পষ্ট ইমেজ এবং খুব মানে
ইমপ্রেসিভ আর কি।
হ্যাঁ হ্যাঁ, এবং যেটা হচ্ছে বলতে পারো যে প্রচ্ছন্ন ইমেজ আর কি, ওনার একটা প্রচ্ছন্ন ইমেজ ছিল। আর সেটা আমার মানে মনের উপর বা জীবনের উপরে বেশ প্রভাব ফেলেছিল। তারপর আমার কলেজ জীবন শুরু হয় করিশ্চার্চ কলেজ থেকে, ওই হেদুর মোড়ে যে করিশ্চার্চ কলেজ, সেখান থেকে আমার শুরু হয়। আমাদের সময় তখন তো ওই ইন্টারমিডিয়েট সায়েন্স 2 বছরের ছিল আর কি। ন্যাচারালি স্কুল ফাইনাল পাস করার পর আমি ওই ইন্টারমিডিয়েট সায়েন্স পড়ার জন্য করিশ্চার্চ কলেজে ভর্তি হই আর কি। সেখানেও আমাদের যে প্রিন্সিপাল ছিলেন তিনি একজন ব্রিটিশ ছিলেন, জন হ্যারল্ড টেলার, এবং তিনি ফিজিক্সের টিচার ছিলেন, বুঝেছ? ফিজিক্সের টিচার ছিলেন। এবং সেই সময়ে করিশ্চার্চ কলেজেতে এই ধরো ফিজিক্স, কেমিস্ট্রি, ম্যাথ, বায়োলজিতে যে সমস্ত শিক্ষক পড়াতেন তারা খুব স্বনামধন্য ছিলেন, এবং তাদের লিখিত বই পাঠ্য ছিল ফিজিক্সের বা বায়োলজির, কেমিস্ট্রির, এবং সেই সব বই আমরা পড়তাম, এবং খুব সেই সব বই খুব পপুলার ছিল সেই সময়। এবং সেই সব টিচাররা আমাদের ক্লাস নিতেন। এই জিনিসটা একটা আমার কাছে খুব উদ্দীপনাময় ছিল আর কি, যে যারা সায়েন্সের বই লিখেছেন, ফিজিক্স, কেমিস্ট্রির মতো সাবজেক্ট, বায়োলজির মতো সাবজেক্ট, তারা আমাদের পড়াচ্ছেন। এটা একটা আমার কাছে একটা খুব মানে মানে কি বলবো, ফিলিং ছিল, একটা উদ্দীপনার উপস্থিত করেছিল আমার ভেতরে আর কি। এবং সেই সব শিক্ষকদের লেকচার যে শুনতাম, বা ওনারা যে সমস্ত তথ্য উপস্থাপনা করতেন তাদের লেকচারের মাধ্যমে সেগুলো খুব আমাকে আকৃষ্ট করতো। এই এটা একটা একটা অংশ। এরপরে আমি যখন ওই স্কটিশ কলেজ থেকে চলে গিয়ে প্রেসিডেন্সি কলেজে ফিজিওলজির অনার্স নিয়ে ভর্তি হই, তখন তো ফিজিওলজি ডিপার্টমেন্টে সঙ্গে যুক্ত হই প্রেসিডেন্সি কলেজের। তো সেখানে যে সমস্ত শিক্ষকরা ছিলেন তাদের সংস্পর্শে আসি আরেকটু ঘনিষ্ঠ ভাবে। ইন্টারমিডিয়েট পড়ার সময় যতটা ঘনিষ্ঠতা হওয়ার সময় তার চেয়েও একটু বেশি ঘনিষ্ঠতা হয় অনার্স ক্লাসে টিচারদের সঙ্গে, ছাত্রছাত্রীদের। এবং সেই সময় তখন প্রেসিডেন্সি কলেজে যিনি হেড ছিলেন প্রফেসর অজেন্দ্র কুমার মুখার্জি, উনি সদ্য ইংল্যান্ড থেকে ফিরেছেন। হ্যাঁ, এবং তারপরে তোমার প্রফেসর সুকুমার লাহিড়ী, উনি অক্সফোর্ড থেকে পিএইচডি করে সবে ফিরেছেন। তাই এই সমস্ত শিক্ষক, তারপরে ধরো বায়োকেমিস্ট্রি যিনি পড়াতেন যারা, প্রফেসর হরিপদ চট্টোপাধ্যায়, ওনারা তো এখন আর নেই, প্রফেসর দেবজ্যোতি দাস উনি এখনো জীবিত রয়েছেন, এবং আরো অন্যান্য টিচার প্রফেসর প্রফুল্লচন্দ্র কর্মকার, রবীন্দ্রনাথ, প্রফেসর রবীন্দ্রনাথ সেন, এরা তখন ওখানকার শিক্ষক ছিল। আমরা যখন ছাত্র ছিলাম যখন ওই কলেজে, তাই এদের ক্লাসে ওই লেকচার দেওয়া এবং পরবর্তী সময়ে প্র্যাকটিক্যাল ক্লাসে যে গাইডেন্স দেওয়া, এইগুলো খুব আমাকে আমার উপর প্রভাব বিস্তার করেছিল। বিশেষ করে এই বায়োকেমিস্ট্রি যে আমার বেসিক ফাউন্ডেশন আমার যেটা হয়েছিল, সেটা কিন্তু ওই প্রফেসর হরিপদ চট্টোপাধ্যায় এবং প্রফেসর দেবজ্যোতি দাস এনাদের লেকচার শুনে এবং এনাদের গাইডেন্সে, এটা কিন্তু খুব একটা আমার ওই বায়োকেমিস্ট্রির উপরে দক্ষতা বাড়াতে সাহায্য করেছে। এটা বেসিক ফাউন্ডেশনটা গড়ে দিয়েছেন ওনারা, এটা আমি সবসময় বলে থাকি। এবং তারপরে ধরো প্র্যাকটিক্যাল ক্লাসে এই যে প্রফুল্লচন্দ্র কর্মকার, উনি হিস্টোলজিতে মানে মাইক্রোস্কোপে নিজেদের যত্ন সহকারে সব দেখাতেন, এবং এক্সপেরিমেন্ট ফিজিওলজিতে তোমার প্রফেসর রবীন্দ্রনাথ সেন, এরা নিজে হাতে এক্সপেরিমেন্ট করে দেখাতেন আর কি। এই সমস্ত এই সমস্ত শিক্ষকদের সান্নিধ্যে এসে প্রেসিডেন্সি কলেজে আমার এই ফিজিওলজি সাবজেক্টের উপর একটা আগ্রহ জন্মায়, এবং এই আগ্রহটা বেশ অনেকটাই মানে মনের ভিতরে গেঁথে যায় এই ফিজিওলজি সাবজেক্ট সম্বন্ধে ধারণাটা। তারপরে বিএসসি পাস করার পর তারপরে তো তোমার ক্যালকাটা ইউনিভার্সিটির সায়েন্স কলেজের পোস্ট গ্রাজুয়েট ডিপার্টমেন্টে এলাম। হ্যাঁ, সেখানে তখন আন্ডার গ্রাজুয়েটও ছিল, পোস্ট গ্রাজুয়েটও ছিল, কাজে আমরা ওটাকে পোস্ট গ্রাজুয়েট ডিপার্টমেন্ট বলছি। এবং সেখানে এসে শিক্ষকদের সঙ্গে পরিচয় হয়, পোস্ট গ্রাজুয়েটে আরো ঘনিষ্ঠতা হয় টিচারদের সঙ্গে, ছাত্রছাত্রীদের। এবং সেখানে এই ক্লাস, টিচারদের ক্লাস, থিওরিটিক্যাল ক্লাস এবং প্র্যাকটিক্যাল ক্লাসে গাইডেন্স, এগুলো আমার উপর বেশ প্রভাব বিস্তার করেছিল, এবং ফিজিওলজি সাবজেক্ট সম্বন্ধে আগ্রহ আর ভালোবাসা আরো উচ্চতা লাভ করে। তাতে আমি সাবজেক্টটা যে একটা বেশ মানে কি বলবো ইন্টারেস্টিং, এবং অনেক কিছু করার রয়েছে, এখনো অনেক কিছু আনএক্সপ্লোরড এরিয়া রয়েছে, এই সব ভাবনা চিন্তা আমার মনে আসতে থাকে আর কি। এইসব কাজেই বলতে পারো এই সমস্ত অভিজ্ঞতাই অ্যাকচুয়ালি আমাকে এই যে শিক্ষক হওয়ার মোটিভেশন হিসাবে কাজ করেছে। হ্যাঁ, এটা আমি বলতে পারি আর কি।
যে আমি শিক্ষক হতে এসেছি সেটা কিভাবে এলাম সেটা বলতে হলে আমার একটু পারিবারিক ব্যাকগ্রাউন্ডের কথাটা একটু বলতে হবে। আমি খুব সাধারণ বাঙালি মধ্যবিত্ত রক্ষণশীল পরিবারের মেয়ে, কিন্তু আমার বাবা মাইনিং ইঞ্জিনিয়ার ছিলেন আর গভর্নমেন্ট অফ ইন্ডিয়ার উচ্চ পদে কাজ করতেন। কিন্তু আমার বাড়িতে আমার মা, কাকিমা, পিসিমা, আমাদের জয়েন্ট ফ্যামিলি ছিল। ওরা কেউই কিছু পাস টাস ছিলেন না, লিখতে পড়তে পারতেন, বাংলা পত্র পত্রিকা পড়তে পারতেন। তো আমি আমাদের বাড়ির প্রথম লেখাপড়া মানে কি পাস টাস করেছি। যাই হোক, যখন লিখতে পড়তে শুরু করে তারপর আমার বাবা আমাকে, উনি যেহেতু মাইনিং ইঞ্জিনিয়ার ছিলেন, বাইরে বাইরে কাজ করতে হতো, তাই আমায় বেথুন স্কুলে হস্টেলে ভর্তি করে দেন। তো আমাদের কিন্তু কোনো স্ট্রেস ছিল না। আমার মেয়েদের কিন্তু আমি অনেক স্ট্রেসফুল মানে আমি ক্রিয়েট করেছি রাদার বলবো, কিন্তু কোনো স্ট্রেস ছিল না। আমি পড়তাম, খেলতাম, লিখতাম, যা হতো হতো, যেমন রেজাল্ট হতো হতো, কেউ কখনো বলেনি ভালো করতে হবে বা কি। তারপরে পাশ করলাম, পাশ করলাম, তারপরে বিএসসি যখন পড়তে শুরু করব তখন বাবা ব্রেবন কলেজে ভর্তি করে দিল হস্টেলে। তখন ওইখানে ভর্তি হয়ে গিয়ে তো অনার্স পড়তে চাইলাম, তখন অনার্স পড়তে খুব অসুবিধা। আমায় জিওগ্রাফি অনার্স দেওয়া হলো, কারণ আমি ওদের আর কিছু অনার্স ছিল না। জিওগ্রাফি বিএর সঙ্গেই হতো, তো ওটা বিএসসি, ওটাই ফার্স্ট ইয়ার, আমরাই প্রথম বছরের ছাত্রী ফার্স্ট ইয়ার ব্রেবন কলেজে বিএসসি। তো তখন হলো না, কিন্তু সিলেকশন টেস্ট হয়ে গেল ডিসেম্বরে। তারপরে ইউনিভার্সিটি থেকে বললো যেহেতু আমি আইএসসি তে জিওগ্রাফি পড়িনি তাই আমায় অনার্স করতে দেওয়া হবে না জিওগ্রাফিতে, তাহলে আমি পাস করতে পারি। তো আমি বাবাকে বললাম অনার্স না হলে তো আমি এমএসসি পড়তে পারবো না। তখন আমাদের রমাদি ছিলেন আমাদের প্রিন্সিপাল ব্রেবন কলেজে। তো রমাদি এই সায়েন্স কলেজের সঙ্গে শুনলেন যে সায়েন্স কলেজে ফিজিওলজি অনার্স আলাদা করে তখন পড়ানো হতো। তখন সায়েন্স কলেজের সঙ্গে কথা বলে আমাকে রোজ দারোয়ান বিকেল বেলায় সায়েন্স কলেজে দুটো ক্লাস হতো, আমার ফিজিক্স আর ম্যাথমেটিক্স পার্ট ছিল, ওরা ওটা নিয়ে নিতেন ক্লাস। তারপর আমি 1:30টার সময় বেরিয়ে আসতাম, এই সায়েন্স কলেজে এসে আবার দারোয়ান বসে থাকতো, আমায় 6টার সময় কলেজ থেকে বাড়িতে হস্টেলে নিয়ে যেত। তো আমার তো মিড সেশনে ভর্তি হয়েছি, মাস্টারমশাইরা খুব যত্ন করেছিলেন, অমিয়দা ছিলেন, মহালানবিশ ছিলেন, তো ওনারা সব আমাকে খুব যত্ন করেছিলেন মিড সেশন। আর আমার না খুব ভালো লেগে গেল, আমার ভালো লেগে গেল মাস্টারমশাইদের অত মনোযোগ, আমি খুব যত্ন করে পড়লাম। মধুসূদন ঘোষাল উনি আমায় প্রাইভেট টিউশনও দিয়েছিলেন। তারপরে যখন আমি পরীক্ষা দিলাম বিএসসি, রেজাল্ট তো এরকম হবে জানি না, ব্রেবন কলেজে বসে পরীক্ষা দিয়েছি, দেখলাম আমি অনেক নম্বর ফার্স্ট ক্লাস ফার্স্ট হয়ে গেছি। তারপরে তো হলো, এসব তো ভাবিনি। তারপরে বাবা বললো তো তাহলে তোকে আমি এমএসসি ভর্তি করে দিয়ে আসি। তারপরে সায়েন্স কলেজে এমএসসি ভর্তি হয়ে গেলাম। তো বাবা তো খুব কনজারভেটিভ ছিলেন, তাই জন্য ওই হস্টেল নিয়ে একটু অসুবিধা হলো। যাই হোক, তারপরে ব্যবস্থা হলো, সায়েন্স কলেজে পড়তে শুরু করলাম। তারপর ভালো লেগেছিল, মাস্টারমশাইরা কিন্তু খুব স্নেহ করেছেন, খুব ভালোবেসেছেন। প্রফেসর মৈত্র, প্রফেসর মাইতি, সব অনেকেই ছিলেন। তারপরে সরস্বী রঞ্জন মুখোপাধ্যায় খুব যত্ন করে পড়িয়েছেন। তারপরে আর আমারও খুব মনোযোগ পেয়ে, আর ওই বিএসসিতে রেজাল্ট ভালো হয়েছিল বলে ওনারা ভালো করে তাকাতেন। তা আমিও খুব উৎসাহিত হয়ে, আরেকটাও ফ্যাক্টর ছিল, সায়েন্স কলেজ আর প্রেসিডেন্সি কলেজের মধ্যে একটা আড়াআড়ি ছিল। তো যাই করেই হোক, এমএসসি তো রেজাল্ট খুব ভালো হয়ে গেল, টু দি এক্সটেন্ট যে আমার নম্বর সে বছর ক্যালকাটা ইউনিভার্সিটির রেকর্ড মার্ক ছিল এমএসসি তে, সবচাইতে বেশি নম্বর। এমএসসি চারটে গোল্ড মেডেল, বিএসসি তে একটা, এমএসসি তে চারটে। তো এইসব হলো। আর বাবা তো বিয়ে দেবেন বলে সম্বন্ধ দেখছিলেন, তারপরে বিয়ে হয়ে গেল।
Okay, I hope that everyone has enjoyed it. I think I guess you will like the next phase of the entire phase of the interviews that was carried out for for the teachers of the Department of Physiology essentially, but we will make no mistake about it that we will eventually interview all our teachers, okay, and who actually contribute to this physiology fraternity, okay, and with that words of my promise, I would like to conclude today. I hope that we would telecast the entire episode of the interviews initially by Professor Ajay Kumar Chatterjee and also by Professor Dr. Manjusri in our next episode. Okay, and it would be streamed in YouTube. I hope that I always would encourage you to watch it and also to subscribe this our Physiosphere channel of YouTube, okay, for for the live events as also live webinars as also for these recorded events, and eventually we will record some of our educational stuff also in future days. And I must mention some of our working team that who actually contributed today. So I just to name a few. Just hold on. Just give me one. Okay, I just would like to name Shruti, who actually took the technical lead. They are all from the very young, motivated students of second semester. Shruti has taken the major lead here in this this initiative. Also, Sriranjana Mitra Chaudhuri, she was the program organizer and who actually started the program, and also Shomu Prabhu. He actually working on the video recording and editing in future casts, not in this program. And Ishika actually she did all the creation of the poster and all. So what you actually saw in the WhatsApp or in the email. And Mustafizur, he actually was instrumental in motivating his peers. And also Abhinanda was also there, Oishi Mojumdar was also there, Sohini Roy and Saurabh Patro and Shubhanjan Bosu. So they actually will take part in the cultural program. And I think so they will coordinate the program in future and they will do their best of their own and they will show their creativity. And I also must thank my son Shubham, who actually did all this video editing. So and I hope that you enjoyed it like anything. He did it in real professional way. And okay, my huge words of thanks, and also to my wife. She is also from Physiology. She is also my classmate and from the Department of Physiology. Okay, so with these words, I would like to conclude today's program of this Physiosphere, the first program. I hope that you enjoyed it, and I hope you always have a look on it and also subscribe it. Thank you very much. Okay, so see you soon. Okay, have a very nice Durga Puja and the festival days.